All right, thank you, everyone. Thank you for your continued participation and support, continuing your brain power through the afternoon. I hope that everyone's enjoyed the free-flowing caffeine out in the lobby, and other things will be free-flowing at a later time. I'm extremely pleased at the moment to introduce John Griffith, Principal Scientist of the Microbiology Department of the Southern California Coastal Water Research Project. And no, that is not SQUIRT, it's SQUIRP, is what people lovingly will call it in Southern California. I've known John for a long time. This morning, Mark was kind enough to place some years into context. I'm not going to go ahead and do that. But I've known John for a long time. He received his PhD in marine biology and biological oceanography, of all things, in the laboratory of Dr. Jed Furman. And that's at the University of Southern California. So even back in those days, John always expressed an interest in novel technologies, ideas, gadgets, and pushing the envelope. So just to give you a few examples of areas that he investigated that resulted in moving us to the 2000s, if you will, for microbial source tracking, are some of the magnetic bead capture technologies for virus and bacterial capture, and also virus concentration technologies. And that ended up evolving into the microbial source tracking and marker advancement work that's really largely gone on and been spurred onward by the state of California. I also want to mention his continued development as an avid USC Trojan football fan. So John's role in our research community has actually been one that's it's novel and it's unique, because it's not only in promoting and advancing the research methods, but it's really one that is strongly rooted in sharing and as a facilitator and implementer. So there's always been a feel to working collaboratively and trying to build collaborative efforts across nations, particularly in the microbial source tracking field, in order to build our capacity and to continue to reach out and continue to push the envelope. So a recent example that John is going to talk about today is the Source Identification Pilot Project, which is funded by the state of California with the Clean Beaches Initiative. Interesting enough that Southern California has something termed that. And while the study really has the benefit of having multiple co-PIs, it's really been John's vision that turned it into a highly collaborative and international examination of MST, not only use and research use, but real-world application. So sharing information and samples and the approach and the technology and now the standardization of microbial source tracking really has become a reality. In this context with this particular study. And it's also resulted, for those of you that aren't familiar, maybe a few students in the audience, in a series of publications that are really nicely featured in the Journal of Water Research in 2013, and other journal articles that continue to come out now, even in 2014. So John's interest in the land-water interface and his experience in biological oceanography has really permitted Southern California to continue to evolve as a national looking glass, if you will, for water quality advancement and protection. So with his hard-working implementation of rapid molecular methods, in the very near future, it will become a reality that literally hundreds of millions of people recreate at California beaches on an annual basis. So when you start to talk about rapid method and rapid notification and implementation, the topics of public health risk and protection become immediately apparent on orders of magnitude and scale that are not seen at many other beaches throughout the country. So thankful to much of his efforts in the last decade, that kind of advancement is a reality. And so, ladies and gentlemen, I present to you Dr. John Griffith.
Thanks, Rachel. Well, thanks for that fine introduction. I don't know if I deserved all those accolades, but first thing I want to do is apologize. I didn't wear a tie, but I want you to know this is one of my nicest Hawaiian shirts. So. Okay, so I'm actually going to talk about this project. It was actually a $4 million project, so it wasn't chump change to do this. And the idea here is beaches are one of California's greatest assets. And so there's about $12 billion annually in gross domestic product to the state that comes from the beaches. And there's about 300,000 jobs. And so when there's a beach advisory or closure, it's been estimated that that costs the state about $100,000. And even though, fecal indicator bacteria are, have been shown to be a good marker for sewage, not all the exceedances we see at beaches are sewage. And so there's a need to tease out what the sources are. And I don't need to tell you that they're found in the gut of most animals. And they don't regrow in the environment. They just grow there. They like it. Okay, I keep hearing people say they're regrowing, but they're not. They're just growing. Okay, so there's a management need. California has something called the Clean Beaches Initiative. And the Clean Beaches Initiative is actually something that's approved by the voters. It's a series of bond measures where the voters actually said, we will pay for bonds to clean up the beaches and improve water quality at the beaches. And mainly this is through infrastructure projects, okay? So BMPs, things like diversions. And it's been really effective. But the obvious problems have been corrected. So what we call the low-hanging fruit, they're gone. So known infrastructure problems, folks wrote proposals and got grants and fixed those. And they've been very effective. And another thing that we've done, because this year we had historic drought, but typically we don't get any rain events probably end of April through the end of October. Maybe one, and that's a really random event. So if you take all the runoff that's coming off the land and you build a sump and put a pump in it, or build a weir across a storm drain and pump that to the sanitary sewer, for the most part in the urban areas, you're taking care of the problem and the beaches clean up quite nicely. The problem is the places where there's a stubborn problem and these diversions have been built, for example, and you still have a problem, they're not so easy to fix. So there was a big opportunity that we saw, and that's the fact that, even since the first MST comparison study we did back in the early 2000s, molecular biology has exploded, and the number of available MST markers has exploded along with it. And the problem is the beach managers don't know what to use. And so which ones work best? Can use them in combinations. How many samples do I need? Basically, what was happening was the Clean Beach Task Force, which is the group that actually approves proposals for this Clean Beach initiative, we'd get these proposals and they'd be so bad that we'd actually have to go out and help the folks develop their proposal to make it good enough to improve. And so the problem was that the idea about what could be causing the contamination was really just a best guess. So it was kind of like, ready, shoot, aim. And so it wasn't working. And so what happened was there was a proposal to take some of these CBI funds and put them toward research to actually develop a protocol that would help people develop successful proposals. Because what they really would like to do is spend these monies on improving the infrastructure that's generally responsible for these problems. Okay, so I didn't do this alone. SCORP was the lead agency. But Allie Boehm, Trish Holden, and Jenny J were all my co-conspirators in this. And as you'll see, we brought on lots of other co-conspirators as we went along. So the project approach. The first thing is we need to know which methods work. So one of the things that we found with the proposals we were getting is typically a municipality would go hire a consultant to help them develop a plan. And the consultant would go to whatever local laboratory they usually worked with, and whatever method they ran, that's what would be put into the proposal, and then that's how the study would go forward. And we had, no idea which of these methods was most appropriate, especially in California, because lots of times it's, something that's published and someone brings it into their lab. We just don't know. So the idea was to develop and demonstrate the source identification protocols at 4 priority beaches once we had identified a suite of good markers, and then prepare a manual that folks could use to actually do these studies in a way that was cost effective. Because, lots of times if there's a big budget, scientists like myself with all kinds of new toys, we go out and measure every darn marker we can, right? I mean, as long as you have the budget, why not? Well, the problem there is that, it's not cost effective, and very often you miss things that are obvious. Okay? And so then finally, the last thing in this project was to actually transfer the technology to local labs. Okay, so why did we need a method evaluation? It should seem obvious, but there are a whole lot of candidate methods, and it had been about 10 years since a comprehensive study had been done. The last study that we did, most folks were doing basically library-based methods, things like ribotyping or antibiotic resistance typing. And there was really only the Kate Field's lab was doing the HF183 marker with endpoint PCR. And that one actually blew everything else out of the water. It was the only one that could identify human and not have a false positive in our previous study. And it turns out that that's still a good marker now. However, just like before, most of the new methods had been developed and tested in one laboratory. And so there's limited geographical evaluation. We weren't looking at how often they cross-reacted with other sources. And you didn't have any comparative studies to find out which ones, if you have this whole, say, suite of human markers, which ones work best. And so that's why the evaluation study was done, and it gave us a basis for recommending the best ones. So our approach was we developed 64 blind samples, and they had a combination of 12 different sources, which I'll show you. We had 41 different methods, 27 participating labs, and we had most methods run by multiple labs, and that's because we needed to know whether or not everyone could do it, not just the method developers, right? And so we had standardized protocols for those labs as well. And then finally, we needed to know, can we transfer it? So here are the sources we put in. We had 3 human sources, actually. We had individuals, sewage and septage, dogs, gulls, cattle, pigs, horses, geese, and these were individual in the case of most of the mammals, but with the birds, the only way we could get enough poop is to composite them. And so we sent a small army of students out all over California, and so just for scale, should know it's about 500 miles from San Diego up to the Bay Area. And this was all done in 2 days, so we had folks scurrying around to different places. We even went into Bakersfield. Any of you that have drove up the 5 have smelled this place called Colingo. It's just a huge feedlot. You need to know that it's there until probably 5 miles before you get there. Okay? And so we all brought them back. All these samples were brought back. And so it turns out that human subjects were the biggest problem, getting individual human fecal samples, because at the universities, human subjects committees are really flinchy about things that are considered, body fluids. So we got around that. I don't have any human subjects committee at my place. And so we had the graduate students carry their samples personally until they got there. And then they were given a container and they did a direct deposit. And so here's a couple of my staff. This is Blythe Layton and Meredith Wraith. And they're sorting all the poop samples from everywhere. Okay? So, to create the samples, we basically combined them with artificial water. So we stirred them if they were liquid, like sewage, or we homogenized them in a blender and screened out the floaties with a 300 micron mesh. And then we measured enterococcus by qPCR as fast as we could to try to give us a metric of how much to dilute them and try to get parity between the samples. Turned out this is pretty hard to do, but that was our initial idea. Then we took equal volumes of each sample and combined them to create a composite. So say with humans, for example, there were 12 individual human sources or samples. They ended up in one sample. All right, and then when we took that, we mixed them up and diluted them out. So this was no small undertaking. We had 6,000 individual filters or water samples. We had 7 days of near continuous work. Pretty much everyone hated me by the end. And then we distributed them between 7 states and 6 European countries. And if you ever need advice about how to get poopy water through customs, turns out I'm pretty good at it. Okay. So we weighed things out, and we measured the poop. This is a blender crew, some of them. These folks were actually undergraduates. They were all into the Tyvek suits and the face shields. By the time we got to the graduate students, they were like, gloves are good. Okay, and see, this is really early on. It's a little pixelated, but you can see they're happy. This is probably day 2. They're still smiling. And I should tell you, we were shuttling students from the airport and back. I had students from Stanford flying in and flying back out. And we just used them until they couldn't stand up anymore, and we put them back on the plane and send them back. Folks driving back and forth from UCSB. I had folks crashing on pallets of boxes. And then by the end, we had no choice but to use child labor. You think I'm kidding? Okay, so if you look around the room, you'll see that there are quite a few participants that ended up being my partners in crime. And I don't really want to go through this list, but what I do want to say is pretty much anybody that does source tracking that's developed methods participated in this study. So we had really good coverage across the US and Europe. So we had different classes of methods. You had presence-absence methods, so single source, binary answers, quantitative methods like the qPCR methods, and then community methods, things like phylochip and TRFLP, so community-type binning methods. And we had several pretty simple criteria. Could they actually say presence-absence? This turns out to be harder than it seems. Can they correctly identify the dominant source or relative contribution? No one can do that well, I'll tell you up front. Repeatability, and then do different combinations provide more information than a single assay, and which combinations work best. And so here's an example of how we looked at them. So this is a binary assessment, and you'll notice we assessed all the methods in a binary fashion so that we could compare them. So that was our one way to compare all the methods. And so what you have here are the different methods. So these are markers, but here's a microarray method. Here's TRFLP. Here's a Bacteroides TRFLP that Yiping on my staff developed. And so what we're looking for is to have them almost 100% sensitive and also 100% specific. And so this assay did really well. Things with an orange dot, the way to read this is that that's greater than 80% sensitive, and all the way over here would be 100% sensitive. You can see this one is really sensitive. It picked up all the samples that had pig in it, but it also picked up quite a few that didn't. So what you want are them to be both sensitive and specific. And you can see the phylochip actually did very well in that regard on pigs too. So here's a binary assay if you look at everything. And you can see here the human assays up here. And we've got several that are pretty sensitive and pretty specific. We have one outlier lab here on the endpoint PCR. But look at the viruses. They're very, very specific, but they didn't detect very many samples. So if they detected a sample, that was good, but they had trouble picking up samples at all. Okay? And then things like phylochip, they were spotty on some of these things. So when we had quantitative assays, then we looked at them like this for sake of this. And all this stuff is in Ali Baim's paper. So if you want to look at more of this, you can actually look at her paper for a general overview, or Blythe Layton's paper for the human assays more specifically. There's a ruminant paper that's out. So if you want to get into the details, they're there. But what we want is we compared them to Enterococcus. So this is how many Enterococcus there are. And then this is how many log copies of the marker. You can see the pig assay, this line here is the limit of detection. And so what we want are everything up here, highly detectable when there's a lot of Enterococcus, but in the other samples, you don't want anything. And so you can see this assay did really, really well. Okay, so how about human assays? Well, you can see the HF183 TACMan did really well. Here's our limit of detection, and a good number of the values are up in the limit of detection, and that's good, so it's quantitative. and we have very few hits on anything but human. But when you look at, say, something like HUM-M2, it's also very, very specific. So, no false positives, but the sensitivity is much, much less. Okay? So what did we get out of this? Well, we basically got a suite of markers that did pretty well and that we thought we could use. And in addition, for the things that we didn't have markers for, say like deer, things like phylochip or the community methods did great. And also the same for chicken. They were able to pull that out pretty well. So we ended up with, a bunch of methods that were specific and sensitive for 5 key fecal sources. But more than that, I think the biggest thing to come out of this evaluation study is we actually had consensus among the scientific community. You never get that, all right? And we brought everybody to SQUIRP, and everyone looked at the results together, and almost every key scientist in the field participated. And Rachel mentioned the water research issue. This is 14 papers, really an entire journal, based on the results of this study. So a consensus that is really rare. So now we have the markers. We moved on to demonstration projects. And so what we really needed to know was, how do we deploy these in the field so that we can tell others how to use them? So we had 4 teams, and we all selected a beach bummer. So we have Heal the Bay in Southern California, and they actually have a beach report card that they publish on a rolling basis every week. And so beaches that are over the year that are graded with an F are considered a beach bummer. And NRDC also has a list of beach bummers. So we all picked a beach bummer. Stanford picked Cowell. UCSB Arroyo Burro. UCLA got Topanga, and we got one of my favorites, Doheny State Beach, just because I like to work there so much. And so the goals were apply and refine the protocols, see if we could find the problem and actually generate a grant proposal from the beach owner. And we were actually successful in some of that. So what did we find? Well, we definitely found sources. So at Cowell, the suggestion had been, the initial hypothesis, is this beach is impacted by kelp. It gets a whole lot of kelp blown up on it, and folks thought, well, the Enterococcus is growing on the kelp because we can measure it when it's there. And it turned out that, yeah, the Enterococcus was growing, but when you actually did a mass balance and you looked at the amount of kelp and the amount of water, there was no way you could account for all of it. Okay? So it turned out to be a minor source of indicator bacteria. The main sources were the birds that were actually picking the arthropods off the kelp and pooping, and humans. And so once kelp was eliminated, we started looking with the source tracking markers, found a buried storm drain that wasn't apparent, and it turned out that there was a cross connection up above. There was a damaged pipe, and it was discharging sewage through the sand. Okay? At Doheny, Doheny Beach was actually one that was a focus of our EPI studies, and there have been a couple of papers out, Shannon McQuaig's paper and Dave Love's paper, about detecting viruses there. And we didn't detect them enough in the EPI study to make any correlative relationship with illness, but we detected them enough to be worried about it. So there's a huge population of gulls there. And these gulls migrate back and forth. In the wintertime, there are probably 100,000 gulls. But during the time we were doing our study, probably on the 1,000 gulls that are resident, and they go back and forth from the dump. Okay? They were a big part of the problem, but there was a big human signature. And what we found is, even though everyone was saying their sanitary system was tight, when we put rhodamine dye in their toilets in the state park, it turned out it came out all over the beach. Now, you couldn't see it with your eye, and they'd dye tested before. They put dye in there, and they'd look for it, and they'd have, a couple rangers standing around. They're like, nope, don't see any dye. We're good. Well, it turns out if you use a fluorometer, you can pick up the dye pretty well. And so they had a persistent problem in groundwater, and we think that that's where the viruses were coming. So the bacteria were getting scrubbed out. We were getting low levels of human markers, but we weren't getting, really smoking hits. When we put the dye in, which is much more likely to travel like the viruses than the bacteria, what we found was it came out everywhere. And then Arroyo Bureau, if you saw Jared Irvin's poster today, he had more detail on this, but the initial idea was it was dogs on the beach. And it turned out the source markers confirmed that the dogs were a primary source, but the problem was up in the watershed where people were throwing their dog poop over the fence and not taking care of their dog waste when they were out walking their dogs, and that was actually coming down the stream to the beach and the lagoon. So what did we learn? Well, the first one was you need a toolbox approach to do this. And so the new genetic tools are great, but some of the traditional tools are also good. And doing your homework and looking at the infrastructure, looking at the historical fecal indicator bacteria, getting out and walking the watersheds, doing things like dye testing and having folks put cameras in. And I ought to mention at Doheny, because the state was having a little monetary issue, state parks ends up being kind of the poor stepchild. And the folks at state parks had suspended their contract with the local sewage agency to monitor their pipes. And so they hadn't checked their pipes in a long time. And so when the dye came out, they said, it's not us, can't be. And then, interestingly, they came up with money to reinstate their contract, and when they put cameras through their lines, they found major breaks in their gravity system. So, it's a due diligence thing, and finding out what kind of maintenance is being done and when it's been done is a big deal. Often, simplest answer is the right one. So we start by looking for leaky pipes, because that's what we found at most places. The thing is infrastructure that's more than 50 years old, that's made of cast iron and is constantly subjected to saline water, doesn't hold up too well. Oops. Last one is get everyone in the same room. It was very revealing. We had, say at Doheny, 5 different agencies that had some responsibility for parts of these systems. None of them really talked to each other, and they all pointed the finger at each other. But what was interesting is when you talk to the folks in the field, they would tell you that, oh yeah, well, we uncovered that force main when we were doing some construction a few years ago, and it started leaking. So we put a band clamp on it, and we covered it back up. Well, no wonder that the whole pipe's like that, right? But these things don't come out unless you get everybody in the same room and you actually talk to them. And it wasn't really, we got them in the same room first, but until we had some data and we started saying, hey, we're finding human marker here, and we find there's dye here, then all of a sudden people had a lot of information to share, mostly about the other folks, right? Okay, so this is an example of Doheny. It has San Juan Creek coming down here, and I just want to show you how complex this is. This was put together by my GIS people from the 5 different agencies. So State Parks is responsible for their little system here, and the sewer lines are in red, and the dots are force mains, and the blue are storm drains. Okay, and you have City of San Juan Capistrano up here. Dana Point has this part. San Clemente's down here. The county runs the harbor. This is their stuff here. And then there's actually a treatment plant right here, and they have their outfall going out under the creek. And so really none of these folks were talking, but they were all blaming the seagulls on the beach for their problem. Okay, it took our GIS person probably 4 or 5 passes to get all the data from these folks. They're all supposed to have GIS data on their systems. But they don't necessarily have it unless you really squeeze them. And they actually got pretty mad at me. One interesting thing we found was, so we have restrooms here, kind of the ends of these pipes in Doheny that we put dye in, and there's one over here inside the park, probably the farthest one from the beach. But when we put the dye in, the first place we saw it was right here. And that was one of the things that the folks said, it couldn't be us. Absolutely can't be us. Well, it turns out there's this storm drain that comes from the Doubletree Hotel. In fact, I think some of you were at a meeting with me there some years ago. And And it comes across the street and it comes out to the beach and it's buried in the sand, so you can't see the end of it. Well, this force main right here runs right across it. And when we put the dye in, what it looks like happened is it came down the outside of this pipe right onto the beach. And the reason they were saying it wasn't us is because, and it couldn't be our system, is it didn't make sense to them that the restroom farthest from the beach was the one that was affecting the beach. But it was just going down the other infrastructure because it was on a gravel bed. Okay, so what came out of this is a source identification manual. So we synthesized everything that we had learned from doing these studies and made it into a document that's available pretty much for anyone. You can go to our website, which is down, actually, this is a link down here at the bottom of my talk. But if you just go to our website, you can find it under publications. And it's basically a tiered approach, starting with less expensive methods and doing all your homework first before you get out there and spend money on source tracking markers that may not tell you what the problem is. We had extensive outside reviews of the Clean Beach Task Force, the Beach Water Quality Work Group, which is a group of state and local representatives of stormwater agencies, health departments, municipalities, NGOs, sanitary sewer operators, that all gets together and advises the state on beach water policy. They reviewed it, and then we also sent it to the folks at EPA. It's hypothesis driven, so you start with a hypothesis and you actually test it. And you make sure you have the right hypothesis and you're not afraid to change it if you're wrong. You start with the less costly methods and then move on to the source tracking markers. And if the source tracking markers don't work, then you go to the community analysis things like the phylochip or sequencing to try to tease out what your issue is. Okay, so once we had the manual, then we were on the hook to train local labs. And so we actually trained a lot of labs throughout the state. And they performed the method for Enterococcus, which didn't require a DNA extraction, which is a little more high-tech for folks that haven't done it before. So we started with that. And then they did the HF183 marker. So we had varying levels of familiarity with molecular methods across there. Some folks from health departments did it all the time. There are some folks that had never done it. And a mix of government agencies, research labs, and consultants. They had a day of training and pipetting proficiency, so Artel actually came out and showed them how to pipette. And it was actually really interesting to see the folks that thought they knew how to pipette, that did this stuff all the time, that didn't do well at all. And then we had some hot shots that did well from the start. And they got gold medals at the end. But very, very revealing and a big source of variability. Day 2, hands-on training, and they did the Enterococcus method, and then they did a DNA extraction, and then the next day they analyzed their own samples, and then we spent the last half of the day on data review and management. And the goal was to get them feeling confident with running the method independently. And then after they went to their home labs, we kind of served as a help desk, and we even developed a web tool, because what we found in our previous studies is about 15% of error is reporting error. People put the things in the wrong Excel column, they calculate wrong, they transpose a number. And so what we did is we developed a web tool where they could take basically the data dump from their thermocycler and put it into our web tool and submit it, and then we had an R script running in the background that would spit out an answer. And so we basically took the calculation away from them. And it had several metrics for quality control as well. And they didn't really have to understand it. What they had to understand was whether they were getting a good answer or not. And then we, later on we explained to them what was going on. So that actually took quite a bit. Okay, then we trained a bunch of agencies. And so you can see we had folks from sanitation districts, cities, public health labs, let's see here, even water utilities. So folks came down from San Francisco and Santa Cruz, Monterey Bay, NOAA, and even a consulting lab. And they all participated in the training. And then 8 of them did an intercalibration with us. And this was important to us because a lot of these labs wanted to participate in our regional monitoring, and we needed to know we had parity of data. And so only labs that could demonstrate they could do the method were we going to let run samples. And so it was a big badge of honor for them. So 8 of them participated. We gave them 6 weeks, and we sent them a care package where they could practice in between and get better, and we provided support. They ran qPCR for Enterococcus and HF183 on blind samples that were duplicate spike text samples and blanks, and then we evaluated on whether they could produce high standard curves, and whether they could accurately measure target concentrations and the unknowns. And then the biggest deal was a lot of these folks are novices. Could they keep from contaminating the blanks? So they did pretty well, and most have been cleared to analyze samples, but we're providing support to a couple that underperformed, and we've actually figured out, we think, what's going on. With one of them, they didn't know how to handle the standards, and so they froze them and thawed them too many times, and they killed them. And then the other one had a systematic error. They were doing something with pipetting where they were getting the same slope as everyone else on their standard curves, but they didn't have the same y-intercept. So, but by the end, they could actually all run the HF183 marker. So we wanted them to be within this level of variability, and these are 3 different concentrations. You can see on their duplicates, they did great. And so we were really happy with that. So what are we doing now? Well, the biggest deal with this is going to be being able to automate it, to reduce human error, but also to deploy it into the field. So many of you have seen this before. This is Mbari's ESP, Environmental Sample Processor. And this is the Microfluidics Block. This is actually the PCR machine right here. It's only a little bigger than the size of this clicker here. So this was developed by Lawrence Livermore and Mbari, the Monterey Bay Aquarium Research Institute, that partnered with us. And we've actually deployed this in the field. So this is Ocean Beach Pier in San Diego. And we are doing an epi study of surfers in wet weather down there. And we wanted to know what's the exposure of surfers where they actually surf. So we put this instrument running Enterococcus and HF183 on the pier, and it was convenient for us because they had a nice cage right here. Turns out the pier is below grade for the sewer system, and so they have to have a lift station, and they had a nice little spot for us right there. So we co-opted that and ran a pipe with a pump down to the surf zone. Had to have divers put that in, and we're able to sample in real time until the ocean busted our pump up. But we're pretty optimistic. We could have actually cored a hole through the pier and put hard pipe in if we'd had a little more time. I think it probably would have lasted. I should say when the waves get big, they actually overtop this and move concrete benches around. So it wasn't really the fault of the installation, but it needed to be built like the rest of the pier to withstand it. So what are we doing now? Well, we're cooperating with Monterey Bay Aquarium to develop a second generation ESP. This is going to be a mobile device, and it's going to use digital droplet PCR. And those of you that saw Yiping's talk know how powerful that looks like it's going to be. Looks to me like we're going to be retesting a lot of these source tracking markers that we just finished testing using digital PCR. But we're developing it to be able to put it in a vehicle, or my boss keeps saying a suitcase model. I think he's dreaming. But I think we can put it in a Jeep, okay, and go out in the field and test MST markers or even measure water quality in real time and telemeter it back to the lab. Monterey Bay Aquarium's endpoint, though, is they actually want to put it in an AUV. Okay, and that would be great to be able to sample offshore, and has all kinds of applications for my agency, looking at distribution of sewage plume and those kinds of things. And because it's actually self-propelled, unlike our Slocum glider, it's unlikely it will be beached and cost us $30,000. So we got a little ambitious and got too near shore. Okay, and that's it. You have to wake up.
Wow. I have a question, John.
Okay.
What were the results when you deployed the apparatus off the pier?
Oh, we got a couple of detects on the human marker, no detects on Enterococcus. But it was sampling every day at 8 o'clock, so that's basically was the regimen we were using to sample on the beach too.
How far out was it? Like how far off the shore?
Probably 50 meters. Where most of the surfers, it was where the waves were breaking most of the time. Because the thing about surfers getting exposed is they don't get exposed, when they're sitting out there on their boards outside of where the waves break. The places they get exposed are paddling out when they're duck diving under waves to get out, and then when they either voluntarily or involuntarily get off their boards, right? And so we're actually working with Mbari to develop an autosampler for our study, because what we really want is for the sample, the personal sampling device on them that activates when they hit the bottom and they go, oh! That's our goal. When they swallow the water, we want to swallow the sample in our sampler. So there are ways to get it when they're submerged. And so we're working on that.
I was looking at the low sensitivity of the virus markers for the human, and I was thinking, could it have to do with the low number of people that contributed to the poo in the sample? But if I look at the data from Europe on, for instance, human adenoviruses in sewage and in recreational waters that are affected by sewage, you almost find human adenovirus everywhere. So could that be an explanation?
It could be, although we had a number of sewage samples, and what we believe has happened is that basically the dilution of sewage was just too low. Because typically we will filter a much bigger volume to look for viruses. They were all treated the same, so they had 100 mils of the sample filtered through them. So it may not have been a fair test for the viruses. But folks wanted to measure them, so sent them samples. In fact, we pretty much sent samples to anybody that said they had a method that seemed credible.
These were really loaded because some of the RNA may have degraded as well. If the viruses were falling apart, the RNA would be really, really loud.
Mm-hmm.
These were loaded. Are there other questions? Did you evaluate whenever you did your analysis multiple markers used at a single time across sensitivity and specificity?
We did try to do that, and we didn't get any good combinations. I mean, basically, the most we got were, human markers, verified human markers, and the cow markers would cluster. But mostly what we found there were markers that did better individually than others. And there were a couple that were pretty much parity with each other.
In this first study, wasn't the intention to normalize to, I think it was about 200 CFU per 100 mil enterococcus?
No, it was 1,000.
1,000. Okay, but still, so you weren't pushing large sample volumes through those filters from the fecal samples and such. So that was another reason why with the viruses we couldn't pick them up, because you would never actually filter that small amount of samples.
Right.
You were doing a virus sample, but the study design was to have all of the samples be consistently processed.
Right.
So none of the samples were particularly processed for viruses, except if they dropped the pH or something like that.
We did find some interesting stuff with viruses, though. It's like the Enterococcus primers picked up enteroviruses in pigs that hadn't been seen before.
Yeah.
So there was enough pig poo there, even though there wasn't enough sewage.
I would say that
And you did a great job.
Thank you. That was all Allie and Lauren.
Great example of a silk purse.
Yeah, with the real-time source tracking PCR on the dock and with the autonomous unit, is there any way to incorporate some type of sunlight data so that you can look at, copy number, but take into account the fact that you may or may not be getting changes in, viable organisms depending on how much sunlight there is?
Oh, sure. We could have put a weather station there. It's just you have to protect it from the local street urchins. That particular beach has a lot of folks that live on the beach. So the biggest problem we had retrieving the instrument was it had been peed on so much no one wanted to deal with it. But the titanium case made sure that the inside was okay.
How do you see regulatory acceptance of source tracking in California? Since you have a guide document, there's no national standards still. I mean, do they use data? Do they accept it? Can they make management decisions based on it?
I think they're certainly it's certainly affecting management decisions. But as far as having any kinds of standards, we don't know the degradation rates compared to the indicator bacteria or the pathogens. So that's actually a study that's just spinning up now that we're doing. And until we know that, we can't even put any parameters on it. And that makes it really hard because, it's just like the beach standards, So everyone in this room knows that there's confidence intervals around those standards. But when it goes into a regulation and the attorneys get a hold of it, it's one number, right? So above that number, you post. Below that number, you're good. Well, we know that that's a bunch of hooey, right? But that's how regulations are. Because folks, if they're going to go to court, it's got to be yay or nay.
Water Microbiology Conference. I know you've heard it already, but if you would please provide us anonymously, not anonymously, with feedback to improve our conference. We have heard a range of great ideas for involving and promoting our great students, undergraduates, graduates, and also postdoctoral research associates alike for ensuing years. I'd like to also send a big thank you out to our sponsors, Aquagenics, BioGX, NSF International, Source Molecular, Thermo Fisher Scientific, and Wells Fargo. And I'd also like to once again thank the advisory committee members, and particularly, who I haven't heard called out by name, but there's a number of staff that have worked tirelessly to put this event together. We truly appreciate their time and their input as well. We've heard very positive feedback about the conference, and we're planning ahead for next year. We are currently planning for the week of May 18th through the 22nd for this meeting. And I'll give you just a few examples of why. You should mark your calendars now. One is that for those of you that prefer yearly to attend the American Society for Microbiology conference, that conference is at a later time next year. It's actually occurring in June. It allows us also to remove ourselves for the benefit of our undergraduate and graduate students from finals week at this university, not only, but also a range of universities across the country, and really allows us to broaden and increase that young student participation. We have a great set of afternoon verbal presentations after this break. So this break is a special one, but not intended to allow you to lose momentum. It's actually 45 minutes, roughly. I ask for those of you that are presenting posters, if you'd stand close to your poster during this longer break. It also might be an opportunity for people that do have to leave to get a chance to speak to you about your poster prior to the afternoon 4 p.m. to 5 p.m. verbal presentation session. The other is that I just want to make the point that we have a lot of posters that are being presented by the people that we that comprise our future in this field. And so I'd like to ask all of your support in supporting them and asking them questions and allowing them to gain valuable input and feedback about their research so they can move their own career advancement forward. So many thanks again. Please put next year on your calendars, and we look forward to seeing you next year.